Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
Go above and beyond.
they become very good at what they do, but only
0.1%.
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
With Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. I have a really great guest today, Robert Siegel. He's a professor at Stanford University in the Department of Microbiology and Immunology. He's going to be a co-author here in the virus book I'm putting together. Very well known. He's been around for a long time. He's part of the program in human biology, the Center for African Studies, the Woods Institute for the Environment. He's been a course director of the infectious disease component of the preclinical curriculum. He's taught many classes, and so extensive background. So Robert, welcome, and thanks for coming.
Thank you. Thank you for the invitation. It's a pleasure to be here. Nice to see you.
Yeah, tell me, what got you interested years ago in infectious diseases?
Well, whenever we tell stories about our past, they're always sort of just so stories that we make up to sort of make our lives make sense. But I think one of the main things that got me interested is that when I was in an undergraduate and when I was a graduate student, they were very, very exciting times for molecular biology and for virology. The very first DNA sequences were coming out. And of course, because they were trying to sequence small things, the first sequence of any biological entity were sequences of viruses. So we were learning about viruses at this, phenomenal pace. And that certainly got me excited. So definitely the times. I've heard lots of lectures on viruses. I do remember lectures by Kathy Dana when I was in graduate school and talking about SV40 and her work in that area. And that got me excited. But interestingly enough, I think my career sort of turned on a conversation that I had with an administrator. I was interested. I had the opportunity to teach a class in the opportunity arose in 1981. And so I actually wrote up two different class proposals. One was on molecular evolution and the other was on human virology. And so I went up to the administrator, who was basically, didn't necessarily know anything about science. And I put them on the table and she said, viruses. And so I submitted the one on viruses and I've been teaching the same class ever since.
Oh, wow. And in addition to teaching, what's your current research about, if any?
Well, I've had a very remarkable career in that I've been able to focus primarily on teaching. So I've done a number of research projects along the way. Currently, I'm working with a number of students on some reviews of the literature regarding aspects of coronavirus. For instance, there's a drug that's an IL-6 inhibitor. And so we've reviewed the literature and we're about to get a publication on that. So mostly my research has been personal. It's been focused on how to be the most effective communicator I can be. So you're always doing experiments when you teach and trying to learn more.
Oh, excellent. Okay. Well, very good. Well, now for the questions. So the first one, do you think viruses are alive or contingently alive when they enter a cell, and why or why not?
Okay, so this is a very interesting question that I spend quite a bit of my the beginning of my course talking about. And I'll begin by saying that I think it's a false dichotomy of saying are viruses alive or are viruses not alive? Because basically I will posit that viruses are sort of on the hazy border between what's living and what's not living. And I often teach with poetry. So this is a stanza from a poem I wrote called The Secret Life of Viruses. And the quote goes, "A virus has a structure giving it properties that lie between inanimate and truly living, less than cell, but more than gene." And the other thing I use when I answer this question is I actually say that many people, when they answer this question, don't define what living is. And so in order to get a really effective answer to this question, you need to sort of agree what you're talking about. And so I actually propose a number of different definitions. So I would say this question is interesting both from a scientific standpoint, because it allows us to sort of understand, what the essential features of living are. It's essential from a pedagogic standpoint because it's useful for me to teach about the nature of viruses. And of course, it's interesting from this linguistic standpoint I was just talking about, namely, how do we define things like viruses? How do we define things like life? So let me pose three different definitions of what's living. The first definition is a definition that came in the late or sort of mid 1800s by two researchers, Schleiden and Schwann, and they propose that all living things are comprised of cells. And for the biological world, that appears to be true for bacteria, for plants, for humans, for everything we can think of. But viruses are not comprised of cells. They're smaller than cells. They actually have to enter into cells and take over in order to make more virus particles. So by that first definition, if we agree on that, viruses are not alive. The second definition, we might say that we could define life by looking at the vital properties, that all the properties that living things have in common. And these would be things like replication, being able to produce more of themselves. And of course, viruses are really, really good at that. But it also includes things like eating and excreting and sensing the environment and homeostasis and movement. And basically, viruses don't do any of those things. They don't metabolize energy. They don't have any mechanism of synthesizing protein or other molecules. And so by that second definition, they fail miserably. So they don't do any of the things that living things do except to replicate. And they do that really, really well. So the third definition actually focuses in on this whole idea of reproduction or replication. And so we can ask, how is it that living things actually replicate? And for this, it's useful to sort of look at the paradigm of Francis Crick, and he came up with an idea called the central dogma of biology. Now, it's got to be important when it's called the central dogma of biology. And basically, what the central dogma does is describes the flow of biological information. And it turns out in all living things, biological information that's passed on to the next generation is stored in the form of DNA. And that information is then actually read out in sort of a Xerox copy in the form of messenger RNA. And that messenger RNA is used to make protein. And of course, in order to be able to pass the information on, that the DNA has to be able to replicate. So all living things have their information in the form of DNA, and they all carry out this process of DNA replication and then expression through mRNA and then into protein. So we can ask, how do viruses comply with that? Well, it turns out that some viruses actually do those sorts of things, and they would be things like pox viruses or herpes viruses, but the vast majority of viruses that infect humans, and I focus on viruses that infect humans, the vast majority of viruses that infect humans, such as coronavirus or flu or Ebola or HIV, violate the central dogma because they either have their genetic information in the form of RNA, or so the majority of them have the genetic information in the form of RNA, but they also violate the central dogma because none of them have both DNA and RNA. They either have DNA or RNA, but not both. So by the third definition, they also fail miserably. But in that sense, it's useful to look at viruses because what we see is that viruses are essentially the exceptions that prove the rule. So in every case, even though they do it in an unusual way, viruses still need to replicate. They still need to copy their genetic information. They still need to produce messenger RNA in order to make viral proteins. And they still need to be able to express that messenger RNA in the form of protein using the host cell's ribosomes, using the host cell's machinery. They're essentially the exceptions that prove the rule of the central dogma of biology. So by all three definitions, they sort of fail miserably. The last sort of bonus definition would be that is it possible to define the minimum genome that it would take to carry out all the functions that we associate with life? And that thought experiment has been done by a number of people, and it's actually been carried out by at least one researcher. And so what they did was they actually took the simplest bacteria, which is a bacteria known as Mycoplasma genitalium, and they eliminated all the genes that were non-essential. And it turns out when they do that, it turns out that there's only about 250 to 300 genes that are necessary to create an autonomously living bacteria. And so and this work was done by Craig Venter. So that basically defines sort of the minimal genome. And again, there's some viruses that have big genomes, but they don't actually encode the right genes to be able to have autonomous life. So again, we come back to this notion that viruses are essentially at the hazy border between what's living and what's not living.
Gotcha. If there's there seems to be a latency period, between which someone's infected and which they get sick, they show pathogenicity. Why do you think that is, and why is that variable under different conditions of different viruses?
Okay, great question. I, just as my teaching role, I want to actually disambiguate the term latency because we use that term in 3 different ways in virology. And one way that we use it is the period of time between when a virus enters a cell and when it starts producing virus particles that are infectious. And so that's one form. Another form is the time between when you're exposed to a virus and when you develop symptoms. And that's often called latency, but it's more correctly referred to as the incubation period. And I think that's what you're referring to here. And yes. The third way that the word latency is used is in conjunction with certain viruses that can actually sort of go to sleep inside the cells. And so the viruses that are really good at that are the herpes viruses. And they almost all go into this state where they could just basically become quiescent and just wait for the right time to replicate. So not all viruses can do that, but certain viruses like the herpes viruses are really good. So let's get back to the question of incubation period. So why do viruses have an incubation period, and why is it different between different viruses? Well, the main reason why viruses have an incubation period is that they immediately start replicating. So there's first there's a lag time between when they enter the cell and when they start producing viruses. And then there's a lag time between when there's only a few viruses and when there's lots and lots of viruses. So generally speaking, they're doing a lot of damage from the very beginning, but it's on a microscopic scale. So we don't experience any symptoms until the amount of damage or the effect of the virus is large enough that it sort of enters our, body consciousness. And so a lot of viruses can replicate for a while before we become aware that we're ill. And there's a second reason why there's a lag, and that is that many of the symptoms that we experience, for instance, when we have the flu, are actually responses to our immune system. And our immune system takes a while before it notices that we're being infected. So when there's only a few viruses there, the immune system's not really geared up high. But when there's lots of viral replication, the immune system is on in high gear, and we start feeling the effects of some of these mediators. Now, there's a second part to that question, and that is that why are some viruses different than others? And what we're going to see over and over again is that viruses really are selected for their ability to replicate and produce more virus particles. And it turns out that viruses have found all different ways of making a living, of carrying out that replication process that are successful. And so just like people have lots of different jobs, there's lots of different niches for viruses to be able to replicate and produce more virus particles. So some of them replicate very fast because they can outrun the immune system. Other ones replicate slowly so they can be sort of stealth and be below the radar screen of the immune system. And that's also an effective strategy. So over and over again, I think it's important to sort of look at the individual virus and try to understand what strategy is that virus utilizing in order to produce more virus particles.
Well, how could there be any strategy if there's no life, no agency, no self-reference, or any of that?
Sure. So the question is, how can something so stupid be so smart? Basically, the answer to that is that these have molecular intelligence. They have evolutionary intelligence. So it's like you have this little computer program, and the computer program is endowed with the ability to replicate, but it doesn't do it perfectly every time. And so if some of the programs actually become better at replicating, they'll take over. And pretty soon you won't see the original program at all. You'll see the modified. And then from there, they can modify further. And as they make each of these modifications and get better and better, it looks like they have a strategy. But what they're really doing is you're just selecting for those variants that are most effective at carrying out that mandate of replication. And the mandate is that basically if things can replicate well, they'll come to predominate and we'll see them. If they can't replicate well, they'll disappear from the population. So there is no sort of will or intention. You're just selecting out those things that are best at this little game, so you can think about viruses as essentially little biological computer program.
When they're inside a cell, they seem to be very capable of taking over cellular machinery and co-opting it for their own uses. Do you think that they're able to make a cell send out specialized extracellular vesicles to communicate with other cells and see the infection status of those cells, or maybe do quorum sensing and kind of coordinate activity.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
So I don't know of any example where that would be true. And so my inclination would be to say they don't do that. But again, the answer to almost everything is if a virus acquired that ability and it facilitated its ability to replicate, then you would see it. So it's not ruled out by being a virus. What's more pertinent and what's more similar to what you're talking about is the response of the immune system. The immune system definitely has the ability to have cells recognize that they're infected, that they're sick, and send out signals to other cells to try to protect themselves. So the cell is smart, the human cell, and the human body is smart, but the virus has the advantage of being able to replicate fast and change really fast. So this is basically a kind of warfare in which the body is being very deliberate about trying to get rid of the virus, and the virus is just selecting for those variants that happen to be able to outrun the body's defenses.
If a cell is intelligent and it can, fight against a virus, communicate that it's in distress, coordinate with the immune system, if a bacteria can take pieces of a virus and, let's say, express viral spike proteins itself and poke holes in other bacteria, then why is it not the same phenomena when a virus co-opts a cellular machinery and changes it to do its bidding?
Well, first of all, I want to talk about the word intelligence. So both of them have biological or evolutionary intelligence. Okay, so they're selected. We get confused because we also use the word intelligent for thought. And so neither one of them has the power of thought, but they both have this amazing sort of like biological intelligence. The difference between a virus and the cell is that the cell is actually the strategy that it has taken on to be able to replicate is one that involves being able to sense the environment and react to it. Viruses only can react in basically an evolutionary sense by being selected. So they don't actually have the same sorts of abilities. And cells, again, don't take up virus components willfully. They take them up by accident, and if it happens to be to their advantage, then they'll be selected for. And in fact, there was a remarkable biologist by the name of Lynn Margolis, and Lynn Margolis came up with a number of very controversial hypotheses, and many of them have turned out to be true. And one of them was that creatures from very distant biological origins exchange genetic information rarely, but when they do, sometimes that can lead to jumps in evolution. So in fact, if we look at viruses, we see that they can pick up genes from their host. And in the same way that hosts can pick up genes from the viruses, what's really going on here is that if it's to the benefit, if it improves the replication of the virus or the bacteria, then that will be selected for.
If I look at three major types of activity, I won't call it behavior, I'll call it activity. You have lytic activity, virus comes in, replicates, blows the cell open. Latency, they'll go in there, like you said, herpes viruses, and hang out and evade the immune system and stay for months or years or lifetimes. And then, in certain cases, HIV endogenizing into DNA and, literally becoming a part of the cell. What do you think governs these three abilities? And especially when a latent virus then, according to host conditions, I would guess it has to be monitoring the host conditions, or the host all of a sudden becomes able to attack the virus. I don't know. Like how would you have these three?
Well, so this again, it's a very interesting question because viruses do such incredible things and have this molecular intelligence. So first of all, the main difference is the difference between different groups of viruses that have been selected for different strategies. Okay, so for instance, the herpes viruses is a family of viruses that uses that strategy to great advantage. Certain other viruses, again, are kind of hit and run viruses, and their strategy is to replicate fast. They get by, and they're very successful without using that sort of more subtle strategy. And their advantage and disadvantage of both. So, for instance, herpes viruses are very large viruses. So they have to carry along a lot of extra genes to be able to carry out this, interesting strategy. In general, again, you're looking for whether they're successful, not just at producing more virus particles, but being able to transmit to other hosts. So, for instance, herpes viruses, as you mentioned, you could be infected with chickenpox, at the age of 6 months, and you could carry that virus along for the next 90 years, and maybe the virus could come out and infect somebody else who'd never been exposed to chickenpox before. So that strategy works to maintain herpes viruses in the population. So you might say that, well, why doesn't every single virus do that? And again, what we see is that viruses use different strategies that are successful. So every virus is not trying to do everything that every other viruses do. They'd have to carry around a big genome, and that would actually select against them. Now, the third group that you mentioned are viruses that actually can stick their genetic information into our genes. And it's important to realize at that point that there's some viruses that, like HIV, that can stick their viruses into our own genetic material. And retroviruses are the only group of human viruses that have to do that in order to replicate. So there's a few other viruses that occasionally do that, but none that actually have to do that to replicate. And again, that's the strategy that they've basically adopted that's successful. It is important to point out that there's a big difference between viruses that stick their genome in the host cell, like HIV, and viruses that end up with their genome in the human genome to be passed on, because those viruses, unlike HIV, have to be able to infect either eggs or sperm or germline cells in order to be passed on. So HIV, even though it gets into the genome of our individual cells, isn't passed on. So we do know that over long periods of evolutionary history, a number of viruses, most especially retroviruses, have managed to stick their genome into the human genome. And in fact, there's probably more DNA allocated, or that's basically has its evolutionary origin as retrovirus than what we might call human genes. So we're more retrovirus in a way than we are human. And again, that strategy has worked for those to amplify up those particular genomes. One issue about that is the fact that the human genome has so much DNA in it that if you think about a computer program that's really, really large and it has some garbage in it, it's usually easier to leave the garbage in, like, what people used to program in a program called Fortran, and there were all these like dead end subroutines, because to actually go through a 1,000,000-line program and actually figure out what's garbage is so much work that people just left them in there. And to a certain extent, that's what's happening with these retroviral genomes, is they're remnants. And in humans, none of those remnants seems to be able to effectively replicate and produce virus particles.
Well, that's probably good. I don't know if we'd want an endogenized retrovirus to suddenly be activated and produce new virions and, do God knows what. That wouldn't be too good.
Yes, that's very good. We do know that occasionally happens in other organisms. So mice have an endogenous virus called mouse mammary tumor virus that actually in some individuals can come out and actually start replicating and give rise to cancer. And there was a hypothesis a while back that maybe since human breast cancer is so common that maybe there's also a human mammary tumor virus, and people look very, very hard and there's just no evidence that we have the comparable virus. And the difference may be that we are very long-lived species. And so if we had something that actually affected our ability to have kids or survive as children, that it would be selected against. So again, it's all about replication and selection.
Well, since viruses can be extremely pathogenic and can kill nearly all of a population, I'm thinking of there's like a rabbit population in Australia where like 99.9% died, but the small remnant then proliferated and they were, immune to the virus, whatever one they had. It seems like viruses can have a heavy hand in evolution and selection and maybe even lead to speciation. I mean, any thoughts on this from your side?
Yes, I love the example of rabbit Mixoma virus, which is a pox virus that basically this rabbit virus was actually introduced for the express purpose of trying to eradicate the also introduced rabbits in Australia, because the rabbits would replicate like rabbits and there'd be lots and lots of them, and they would eat so much of the food they would actually impact the local fauna. So for instance, they would eat up all the food that maybe the kangaroos might eat or some of the other indigenous animals. So this virus was actually introduced for the express purpose of trying to eradicate the rabbit population. So this was a virus that is in its normal rabbit host. It's not nearly as lethal as it is in the European rabbits that were introduced into to Australia. And what happened was quite remarkable, and that is that virtually every rabbit disappeared, and there were no more rabbits. It seemed like there were no more rabbits in Australia. But over time, rabbits began to appear. And now the rabbits and the virus are in this sort of dynamic equilibrium that some people have called a predator-prey relationship. So as there's fewer and fewer rabbits, the virus can't replicate. And so really virulent, really nasty viruses are selected against, because if you kill everything in your little local population, you don't get passed on. Whereas if you are in a population where you have some variants that are a little bit milder that allow the rabbit to replicate, then those are more successful. And when those variants start to arise, there starts to be more and more rabbits. Well, as there's more and more rabbits, that means that now the more virulent rabbits, the ones that can replicate faster and more aggressively, are now selected for. So you have this sort of change in virulence over time in the virus to allow the rabbit population to increase, and then it will decrease. So it's very much like a larger predator, like a lion, that keeps an antelope population in check. These are the smallest predators that are keeping their rabbit population in check.
Well, with endogenized viruses, though, like in humans, I guess they comprise 8% or more of all our genes. And supposedly they're what allow for placental mammals. So, I mean, do you think they also, besides just culling and nearly killing everyone, they push creatures to evolve in certain ways they otherwise might not?
Well, so there's a couple of parts to that question. First of all, viruses, the purpose of viruses is not to cull. The purpose of viruses is their evolutionary intelligence is to produce more virus particles. So some viruses do that really, really well without actually having any clinical effects at all. So there's a widespread virus called TTV, Torquay-Tenu virus, and that virus is found in many, many, many people. And we have yet to find any kind of clinical disease associated with it. So it's remarkably successful, even though it doesn't actually seem to damage the host in ways that are obvious. They don't necessarily, they're not selected for being extreme. They're selected for their ability to be effective replicators. Now the question is, what about things like the ability to create the placenta? Well, it turns out that just like the virus is selected for variants that can help it by picking up host genes, the host has all these retroviral genes. And so if some of them actually confer an advantage, then they'll be selected for over time. And so there's a couple of examples of that. For mammals, it appears that the ability of viruses to fuse cells together was actually was co-opted by mammals to create the placenta, which includes basically cells fused together. So, and another example is that some people feel that some of these endogenous retroviruses may have facilitated human evolution by a remnant enzyme called salivary amylase, which allows bread to taste really good. And so that actually evolved in fairly recent times, along with the cultivation and of farming, the rise of cultivation.
Okay.
So yeah, we will pick up, if we have the opportunity to find one of those endogenous retrovirus or other retrovirus or other viral genes, those will be selected for because they will give their host an advantage.
Well, in terms of immunity, what role do you think our virome or a bacteria's phagome plays? And I'm thinking about cholera. It requires, I guess, a piece of phage genetic material to make it pathogenic to people. So that kind of contributes to its success, maybe a replication, the bacteria itself. Do you think that viruses play a role in our immune system?
So again, I'll kind of cut that into several parts. Well, first of all, one can make an argument that viruses actually gave rise to our immune system, because if you look at viruses, they have two phases of existence. They have an inert phase outside the cell where they really don't move or metabolize or do anything like that. And then they have a dynamic phase when once they enter the cell, basically their genetic machinery starts taking over every aspect of the cell's behavior. If you look at the immune system, it's also divided into two. We have the branch of the immune system called the humoral immune system, and that basically produces antibodies that inactivate extracellular or inert phase viruses. And then we have the cell-mediated immunity or T-cell immunity that is able to detect virally infected cells cells or dynamic phase virus and kill those cells. We don't have a good way of right now of eliminating the virus from those cells, but what we do is we sacrifice those cells in order to prevent those cells from making more virus that can be passed on to other cells. So in that sense, the viruses have very much affected our immune system, which is helpful in all sorts of ways, particularly, for, again, long-lived creatures that have to deal with lots of different things. We can't respond to the environment by evolving really fast. So we have to have, adaptive defenses. Now, cholera is a great example because, first of all, it underscores the need to look at each virus separately. So in the case of bacterial viruses, some of them actually harm the bacteria and some of them help the bacteria. And you can look at this in the way that if the virus can benefit and the cell can also benefit, then that will be selected for. So in the case of cholera, they're infected with a phage called CTX, which stands for cholera toxin, and it encodes the thing that causes the massive diarrhea. From the bacterial standpoint, that's actually advantageous to the bacteria because it allows the massive diarrhea might hurt the host, but it's more likely to spread to a new host because the bacteria is everywhere. So in that case, the virus actually benefits the bacteria. But in many other cases, in the vast majority of cases, viruses actually hurt the bacteria. And in fact, as far back as before 1920, a proposal was made that maybe we could use viruses to kill bacteria and stop bacterial infections with this strategy that my enemy's enemy is my friend. So that actually was used before the development of antibiotics. And now, with the development of antibiotic resistance, people are actually looking back into this idea of using bacteriophage as a kind of therapy for bacterial infections. And this is advancing fairly rapidly for people that have cystic fibrosis that have bacteria that are really kind of untreatable with the current antibiotics.
Oh, that's excellent. I didn't know they were looking at phage for that. That's great.
Yeah, there's a book called Salt in My Soul, and it's about an undergraduate at Stanford who actually had cystic fibrosis, and her sort of battle with cystic fibrosis. She ultimately died of a bacterial infection, but basically she was ultimately treated. It was too late when she was treated, but her story has actually given rise to a renewed interest in this kind of therapy for cystic fibrosis.
If I'm infected with a virus and I label myself number 1, and then I infect you and you're number 2, and you infect someone else and so on, by the time I get to person 100 and its passage through 100 people, what would you expect and how would you expect the virus to change, and how would you expect the 100th person to experience it versus the first?
So again, I tend to be a little repetitive because, the answer to this question again is kind of, evolution. So for some viruses, the virus might become more virulent, and for some viruses and be successful, and some viruses might become less virulent over time. And so we have examples of both of those sorts of strategies. So generally speaking, like the rabbit, or like coronavirus, if we throw a lot of susceptible hosts and the virus can replicate a whole bunch of times, it's much more likely to be more and more vigorous in its replication because those variants would be selected for. So as long as there aren't any barriers to becoming more virulent, the virus will tend to evolve virulence. On the other hand, if there are barriers, there's sometimes you will evolve and become more benign. And again, herpes viruses are great examples, or TTV, because the majority of people that are infected with something like cytomegalovirus have the virus in their body for the rest of their life, but they're not really sick with the virus. So what will happen will often depend on human behavior and the number of susceptible hosts that are present. And by the way, over time, if we look at something like smallpox continued to be deadly even, over centuries. And in fact, it's estimated that more people died of smallpox than any other virus during the 20th century, even though it was eradicated in the 1970s. So it didn't become more and more benign, whereas other viruses that are successful like TTV don't become more virulent. So you have to look at each virus individually and try to sort of figure out what the success of that strategy is for that particular virus. In the current pandemic, for coronavirus. What would be great would be to limit the total number of replications, both in individuals and overall. Take the example of HIV. If we can squash down the replication of HIV in an individual, that individual is much less likely to develop resistance to antivirals. Whereas if we let the virus produce lots and lots of variants, lots and lots of bets on the table, it's much more likely that some of those variants will actually be resistant. And that's the other reason why often it's useful to treat things like tuberculosis, which is not a virus, or something like HIV with multiple drugs, because that decreases the probability that you can develop antiviral resistance, because you need multiple mutations in order to become resistant to multiple drugs at the same time. And that ultimately should be the strategy that we're using for coronavirus. But right now, our armamentarium is pretty meager. We essentially only have one licensed drug that's an antiviral, and that's remdesivir. The other licensed drug is dexamethasone, but that's not an antiviral. That's actually to minimize the impact of immune dysregulation. So it probably doesn't affect the viral evolution.
Gotcha. If I have a choice to get infected, it's a made-up scenario, but if I could choose if to get infected with a virus by someone that's like deathly ill from it versus someone that doesn't really feel anything, what do you expect that I would experience? Would I be more sicker if I get infected from a very sick person or from someone that's asymptomatic, regardless of viral load? And I know my genetics and my experience and my body is unique and all that, but any predilection there, you think?
Yeah, so again, I want to take it on sort of a case-by-case basis, but we can look at some examples. And that is, so generally, if something is more aggressive, it's possible that we've now selected for a more aggressive variant of a particular virus. So if that were the case, it would be better to be infected with a more benign variant. And we know, like for smallpox, there were two main types of smallpox, which we call variola major and variola minor, and they differed greatly in their mortality rate, but they were similar enough that they conferred cross resistance. So if you were infected with the mild strain of variola minor, what's also called elastrum, that would confer resistance to variola major. And that actually resulted in a strategy to stop smallpox. And that is, people would actually inoculate people with mild strains of smallpox in order to prevent them from getting more virulent strains of smallpox. And that was called variolation. And that's basically the precursor to vaccination. So that's one example. But if we take, like, for instance, coronavirus, the likelihood is that the more serious infections are due to host factors. So if you're young, you're less likely. So in that case, it wouldn't really matter which strain you got. What might matter is the number of virus particles you're infected with. So there's growing evidence that if you get a bigger dose, if you get a bigger inoculum, you're more likely to be sicker because it takes longer for your immune system to gear up. So in that case, you may want to, it's not necessarily how sick somebody is, but it's how many virus particles they're producing. So for instance, for coronavirus or for flu, we are often producing more virus particles before our immune system kicks in and before we're really sick. And so you'd be better off to be infected by somebody who's late in their, who's basically been infected with flu for, 10 days than somebody who was infected with flu for just, a day or two, who's now producing lots and lots of virus particles. And if we look at the example of HIV, the vast majority of people who are infected with HIV are infected with relatively asymptomatic individuals who are engaged in high-risk behaviors. The people who are really sick usually are not engaged in high-risk behaviors, and they do not drive epidemics. So it's the people early in infection that tend to drive the pandemic. Now, again, if we look at Ebola, it's a little bit different. What we've discovered in the last few years is that sicker people tend to have more virus particles, and so you wouldn't want to be infected, with somebody late in disease that was really sick. But again, for Ebola, it matters more which strain of Ebola it is than anything else, because some strains are more deadly than others.
Okay, well, very good. Last question. What do you see as the future of virology, and, our understanding of it, over the next few years? And then, what are some big goals that you have for maybe, 20 plus years out?
20 years out. Okay. So as Yogi Berra says, it's really difficult to make predictions, particularly about the future. But if we look at the pace at which people are learning about viruses and learning about biology, it's really remarkable. And I can say that when there's something like a pandemic, that's actually spurs advances in all sorts of areas of science, and especially in the area of virology. So if I were just sort of to extrapolate logically from what's going on now, we are beginning to understand more and more about the detailed molecular, even atomic structure of some of the proteins that are made by viruses. We're understanding a lot more about different strategies, unique strategies that can be used for vaccination or for treatment. A huge breakthrough occurred a number of years ago when some treatments were developed for hepatitis C. This was a disease that was very difficult to treat for a long time, and now we have drugs that can actually cure this chronic infection. So again, as we look at more specialized treatments that are aimed directly at the viral proteins, we're getting better and better at that. I think one big gap in our knowledge that people are looking at, and that coronavirus has sort of underscored, and that is that the relationship between the immune system and viruses. And so basically, we know a lot about the immune system, but the fact is that because these are two interacting systems, the virus is rapidly evolving. The immune system is trying to adapt. So we are learning a lot more about how the immune system deals with viruses, and how the immune system, for instance, changes as we get older. So a lot of what may be at risk for older individuals is a change in their immune system. So for instance, their T cell immunity tends to be less vigorous as they get older, but their innate immune system that produces these cytokines seems to continue to be vigorous. So we're learning lots of different things about this sort of interaction between humans and viruses. The other thing that you sort of alluded to a bit earlier, and that is that using viral tricks to basically our own advantage. And viruses have produced many different kinds of therapeutics. They produce tools for diagnosis, and we can use them for things like vaccination and for gene therapy and things like that. So, even CRISPR-Cas, which is actually part of the bacterial immune system against phage, is now being utilized in all sorts of remarkable ways. That system probably would not have evolved if bacteria didn't have to protect themselves against phage. So again, humans are clever, and they're utilizing all these new tools. Also, we're discovering many, many more viruses, and it turns out there's more kind of genetic diversity among viruses than almost anywhere else in the biological world. If we look right now, there's only been 6,590 viral species that have been characterized. It's likely that there are hundreds of thousands of different viruses that haven't been characterized. And as we characterize them, when we look at their genetic makeup, we're going to, first of all, learn a lot about biology, but we're also going to get tools that we can use for all sorts of interesting things. So the future of the science of virology is kind of unlimited and exciting.
Well, excellent. Well, Robert, thank you for coming, and I appreciate you contributing to this. Your answers are great, so thank you.
It's such a pleasure, and the opportunity to talk about viruses, which I've made a whole career out of. And I think it's an exciting field. And I congratulate you on this project, because I think it's a great project, and hopefully it will educate lots of people about these interesting, fascinating, dangerous entities called viruses. So thank you very much.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.